Science Journal for Kids and Teens presents How Can We Detect Rare Animals? Read by Miranda Wilson. Abstract. Did you know that scientists can use DNA to figure out which organisms have been in an area? Organisms leave DNA behind in the environment. We call this DNA environmental DNA, or eDNA. Using eDNA is a new technique that scientists have used in water environments. We wanted to figure out if this same technique could work to find species on land. We collected soil samples containing eDNA from golden moles in the western part of southern Africa. These mammals are hard to observe because they live underground and some species are rare. People believe that one species, de Winton's golden mole, might be extinct. We extracted eDNA from soil samples. Then we compared the eDNA to DNA samples we collected from golden moles and from databases. We found that all 4 golden mole species live in this region of Africa. That means that we can use eDNA to find rare animals in land environments. It also means that de Winton's golden mole is not extinct. Introduction. Scientists study DNA because it carries information about how an organism will live and function. Over time, DNA has become important in ecology. Ecologists are scientists who study the interactions between organisms and their environment. Ecologists are now using environmental DNA, or eDNA, to study species. eDNA is DNA left behind in soil, water, or sediments. Organisms leave DNA in the environment as hair, fur, skin, urine, saliva, or blood. eDNA can tell scientists what organisms are in an area. It can also help scientists learn more about rare organisms that are difficult to observe in the environment. Most ecologists who have used eDNA study ocean organisms. That's why we wanted to explore the use of eDNA to study land animals. Specifically, we wanted to use eDNA to find evidence of the golden mole. The golden mole is a species of insectivore that lives in southern Africa. They are very difficult to study and observe. That's because they burrow in soft soils. There are 4 species of golden mole that live on the west coast of southern Africa: the Cape golden mole, Grant's golden mole, Van Zyle's golden mole, which is endangered, and de Winton's golden mole, critically endangered and believed to be extinct. So, we used eDNA to determine where each species of golden mole can be found in this area of Africa. Methods. We collected 49 eDNA samples from sites in 6 regions along the west coast of South Africa. We collected soil samples from sites where there was golden mole activity. We identified active sites using a trained scent detection dog. Here in Figure 1, you can see that we identified active mole sites using a trained scent detection dog. There are 2 women in the photo. Both are kneeling in a shrub ecosystem. The woman on the right is holding the scent detection dog, who is black and white. Between the 2 women are some of their scientific instruments. We took soil samples from inside golden mole tunnels whenever possible. We also set up 2 pitfall traps. These traps are a safe way to capture a golden mole. We then collected DNA from the 2 moles that we trapped. We also collected DNA samples from the bodies of 2 golden moles that people's pets had accidentally killed. In the laboratory, we extracted the eDNA from the soil samples and DNA from the golden mole samples. Here in Figure 2, you can see we took the soil samples back to the lab to extract and sequence the eDNA. In the photo is a woman wearing a mask and sitting at a lab bench. She is wearing blue gloves and is holding a micropipette in one hand and a small vial in the other. Then we sequenced the eDNA and the DNA. That means we analyzed each one to determine its structure. There are 4 chemicals that make up DNA. We call them A, T, C, and G. These chemicals are like paper clips that link together in a chain. When we sequence DNA, we learn the order of the chemical links in the chain. When we sequenced the DNA, we focused on 3 sections. That's because each animal species has a unique order of the DNA chemicals in these sections. These unique sections are called a DNA barcode. Once we had sequenced the barcodes for the eDNA from the soil samples, we matched them with the DNA barcodes from the golden moles, like a shape matching puzzle. We also used golden mole DNA from a database to help us. When we found a match in a soil sample, we knew that a golden mole was in that region of Africa. Figure 3 shows the process of using eDNA to identify which species of golden mole live in a region of Africa. The first step, seen in the upper left corner of the image, is a soil sample collected. The eDNA in the pile of soil is represented by different colored shapes. The second step, seen in the top middle, is to do DNA extraction. You can see 4 eDNA strands represented by double-stranded DNA that are different colors, with the corresponding shape in the upper left of the DNA strand. The third step, seen in the top right corner of the image, is eDNA sequencing and barcoding. In this portion of the image, you can see the individual chemicals that make up DNA, represented by A, T, G, and C in the 4 eDNA strands. The fourth step, seen in the middle row of the image, is eDNA and DNA comparison. The 4 eDNA sequences are colored with their corresponding shapes. Below each eDNA sequence is the comparison DNA sequence for the 4 different mole species. From left to right, they are the Cape golden mole, Van Zyl's golden mole, Grant's golden mole, and De Winton's golden mole. The fifth step that can be seen at the bottom left is species identification. In this case, blue DNA with a square is identified as Van Zyl's golden mole, a green star as Grant's golden mole, a red triangle as the Cape golden mole, and a purple circle as De Winton's golden mole. Looking at the diagram, why were DNA samples from the golden mole necessary for our research? Results. From our eDNA analysis, we found that all 4 golden mole species live along the west coast of southern Africa. We found Grant's golden mole and De Winton's golden mole in all 6 regions. In 3 of the regions, we found golden mole eDNA that could come from Van Zyl's golden mole. We also found evidence of the Cape golden mole in 2 of the regions. Discussion. Our results showed that eDNA techniques work in land environments. Plus, they can detect rare species. We now have evidence that De Winton's golden mole is not extinct. Instead, it is present in all the regions we investigated. We also found where the 3 other golden mole species are present. Using eDNA in land environments will help with conservation. Golden mole populations are decreasing because of human activity. Diamond mining is the main reason. To find diamonds, miners move sand dunes on the coast. This means less habitat for golden moles. Knowing where the golden moles live can help people protect them. But first, ecologists need to better estimate the number of golden moles in each region. They also need to find more details about where the golden moles live within that region. Then scientists and governments can work together to develop a plan to protect these species. Conclusion. Environmental DNA is a great reminder that everywhere we go, we always leave something behind. How can we make sure to leave as little behind as possible when we're outdoors? Follow the basic leave no trace principles. These include properly disposing of your trash, keeping your distance from animals, don't approach, chase, or feed them, and leaving behind what you find. Instead of taking home a plant or rock, take a picture to capture the memory. By respecting wildlife and their habitats, you can help conserve the species in your area and in places that you visit. Thank you for listening to this recording. This work has been adapted from the original peer-reviewed paper in the journal Biodiversity and Conservation, published on November 24, 2023. Research conducted by Samantha Maynard, Quobus Teron, and others from the Department of Botany and Zoology at Stellenbosch University and the Endangered Wildlife Trust, both in South Africa. See the full list of authors and their affiliations in the accompanying PDF. Please visit our website, sciencejournalforkids.org, for more free science teaching resources.